The group of reactions, collectively called glycolysis, converts glucose, glucose 1P, or fructose to pyruvic acid in the cytosol. Several reactions of glycolysis also occur in chloroplasts and other plastids, but the complete pathway probably does not. Glycolysis is the first of three closely related phases of respiration and is followed by the Krebs cycle and electron transport processes occurring in mitochondria. The individual reactions of glycolysis, now believed to occur in all living organisms, were discovered by German scientists interested in alcohol production by yeast and by others concerned with the breakdown of animal starch, glycogen, to pyruvic acid in muscle cells. The term glycolysis, meaning lysis of sugar, mean a breakdown of sugar into ethyl alcohol. However, most cells produce pyruvic acid when normally aerated instead of ethanol. Furthermore, the common sugars that are broken down are hexoses, so glycolysis has come to mean the degradation of hexoses to pyruvic acid, although many animal biochemists use the term to mean degradation of glycogen, animal starch, to pyruvate. The individual reactions of glycolysis, the enzymes that catalyze them, and the particular requirements of the enzymes for metal activators are in. The overall process, however, can be summarized as glucose plus to NADP plus to adenosine diphosphate. To yield to hydrogen phosphate pyruvate plus to NADPH plus to hydrogen ion plus to adenosine triphosphate plus to water. Glycolysis has several functions. First, glycolysis converts one hexose molecule into two pyruvic acid molecules and oxidizes the hexose. No oxygen is used, and no carbon dioxide is released. For each hexose converted, two molecules of NAD are reduced to NADH. These NADH are important because oxygen can subsequently oxidize each in a mitochondrion so that NAD is regenerated and two molecules of ATP are formed. Some NADH that do not enter mitochondria are used in the cytosol to drive various anabolic, reductive processes. These NADH produced during glycolysis are important compounds. Inspection shows that NADH is formed only at one stage in glycolysis, during the oxidation of 3-phosphoglyceraldehyde to 1,3-bisphosphoglyceric acid. A second 4-fructokinase. To ATP are later released for each 3-carbon unit involved in the reactions catalyzed by phosphoglycerokinase and pyruvate kinase. Thus there is a total production of 4 ATP per hexose used and a net production of 2 ATP per hexose. If glucose 1P, glucose 6P, or fructose 6P is the substrate, one less ATP is required to begin glycolysis, so the net ATP production is 3 per hexose phosphate. In photosynthetic cells, 3-PGA, 3-phosphoglyceraldehyde, and the hydroxyacetone phosphate produced in the Calvin cycle can become substrates of glycolysis when they are transported into the cytosol from chloroplasts. So you can see that photosynthesis becomes connected to respiration in several ways. A third function of glycolysis is the formation of molecules that can be removed from the pathway to synthesize other constituents of which the plant is composed. Finally, glycolysis is important because the pyruvate that it produces can be oxidized in mitochondria to yield relatively large amounts of ATP. The function of glycolysis is the production of ATP.